Hello and welcome. In this lesson, we look at classification. Now, living things are numerous. In fact, it is estimated that there are over 5 million different kinds of living organisms on Earth today. These forms of life have characteristics that distinguish them and enabled biologists to arrange them in groups. If we look at these example here we have such examples of different types of living organisms from the invertebrates. These are invertebrates, that is, they lack a backbone. These are animals without a backbone. We have the sea urchin, fly, snail, jellyfish, prawn, earthworm, spider, cockroach. These compared to animals with backbones, the horse, cattle, donkey, goats, birds represented by chicken, pigs, and sheep. Organisms also include plants. All these are different types of plants. So you find that there are so many different types of organisms that it will be necessary to group them, to classify them, if we are to make easy study of them. Now, because of the large variety, there is need for classification of living organisms. Now, we start by defining what classification is. What is classification? Classification simply refers to the grouping of living organisms. Grouping of living organisms. And this is usually done according to their structure. So classification is defined as grouping of organisms. Organisms are placed into groups. Now, the science of classification, that branch of biology that deals with classification, is known as taxonomy. And the biologists who study taxonomy are known as taxonomists. Why is it so necessary to classify living organisms? What is the importance of classification? There are a number of reasons, and 3 prominent reasons include: it helps us to identify living organisms. If we have a classification system, then we can use it to identify organisms that may not be so well known to us. We can also use classification to group organisms according to their features, so as to make it easier to study. For example, all arthropods are placed in one group, and the study of these arthropods, and especially the study of insects that belong to this group arthropods, is known as entomology. So by grouping insects into one group, then it makes it easier for us to study them. A third reason for classification is that it helps us to understand the evolutionary relationship between different groups. For example, we are able to understand how we, the human beings, are related to the apes, or how the cattle are related to buffaloes. So by understanding the evolutionary relationship, we're able to take care of different organisms, because you know that by studying the buffaloes, then you're able to deal with problems and diseases that face the cattle which we keep as domestic animals, because the two are closely related and they are likely to be faced with similar environmental problems. Now, when grouping organisms, they are placed into classes or units that are known as taxa. Taxa is plural, and taxon is singular. So, after studying organisms, we place them into units based on their structural similarities, and these units are known as taxa. So, the group for arthropods is a taxon, just like the groups for the primates is also a taxon. It consists of organisms that have a lot of similarities in common. Now, when placing organisms into taxa, we use certain observable features. Now, to clearly see these features, sometimes we need to use a hand lens. We need to use a hand lens to make observations of these features easier and clear. Now, a hand lens is a simple apparatus that is used for observing small objects. It consists of a convex lens mounted on a frame or a holder. At one end, there is a handle that supports and holds the lens plus the frame. Now, the hand lens, enables us to observe objects that will otherwise be too small, such that the image that we see is larger and shows us clearer features. A hand lens can magnify an object up to times 30. In fact, the magnification ranges from times 6 up to times 30 for most hand lenses. So when using a hand lens, we are able to observe features on a specimen that would otherwise be very difficult to observe using our naked eyes. Now, after you've observed, usually make a drawing, and it is important that you learn to calculate the magnification of a drawing of an object that has been observed using the hand lens. Now, to calculate the magnification of our drawing, we use this formula. We measure the length or the width or whichever dimension of the drawing, then we divide by the length of the actual object. So let's say if I was observing an object, let's say a leaf, and then I drew the image of the leaf. So this is my drawing. So to calculate the magnification of my drawing, I measure this length. Let's call it x. So using the formula, magnification is equal to x, say in centimeters. Then I also measure the actual length of the leaf that I've drawn. Let's call it y, centimeter. So for example, if let's say the length of the drawing is 10 centimeters and the actual length of the leaf is 5 centimeters, then this will give me a magnification of times 2. And it is important to use the times symbol to show magnification. So the magnification of this drawing is times 2, meaning that it is 2 times larger in length compared to the actual object. Now, so when we can use the hand lens to observe the external features of organisms, and these features in plants include, comes to the leaves, the venation. Some leaves have net-like venation. So you can see these 2 leaves have net-like venations, others have parallel venation. We can consider the shapes of the leaves. Like here we have this one that is pear-shaped, heart-shaped, one that is round, needle-like. Arrangement of the stem. There are those leaves that are arranged in a circle around the stem, so they're said to be whorled. There are those that are simple. Then there are those that are compound. We can look at the leaf margin. It can either be smooth or toothed or lobed. And then arrangement on the stem can be alternate, opposite, or whorled. So these are some of the features that we use to classify plants. We can also look at the type of roots. Can either be taproot, like in the dicotyledonous plants, or can be fibrous. When it comes to animals, the features that we consider may include, presence or absence of a shell, for example, like on the snail, feathers or fur covering the body. We can use locomotory structures. Does it have legs or does it have wings, like in the fly? Symmetry is also important. Symmetry refers to the number of times that a body can be divided into 2 halves along a particular plane. Like the starfish is said to have a 5-ray symmetry. You can divide it into 2 equal halves along that one plane, the first one, second, third, fourth, and fifth. So it's said to have a 5-ray symmetry. While an animal like the fly can only be divided into 2 equal halves along one plane. So the fly is said to have a bilateral symmetry. So symmetry is also a feature that is used to classify living organisms. And you can also consider the presence of sensory structures like the antenna. Here the squid has an antenna, the cockroach, and the prawn. These have antennas for sensitivity. So those are some of the features that are used to classify animals into groups.